The overall goal of the following experiment is to identify abnormalities in nerve function in patients undergoing chemotherapy treatment using axonal excitability studies. This is achieved by stimulating the median nerve at the wrist and recording the compound sensory action potentials from the second digit or compound motor action potentials from the abductor pollicis brevis muscle. Next, a stimulus response curve is produced to determine the changes in threshold current required to achieve a target amplitude, which is tracked online using threshold tracking techniques. Multiple nerve excitability parameters are recorded in response to different stimulus patterns, which assess markers of peripheral ion channel function and membrane potential in order to evaluate nerve function. Results are obtained that show the longitudinal changes in sensory axonal excitability with chemotherapy treatment, and provide a method to detect these changes earlier than conventional techniques.
The main advantage of this technique over conventional nerve conduction studies is that it provides information about the intrinsic function of the axonal membrane, including constituent conductances, rather than simply amplitude and conduction velocity. Demonstrating this technique will be Dr. Cindy Lin, a senior lecturer in neurosciences, and Dr. Susanna Park, a postdoc from my laboratory.
For this study protocol, patients are typically referred by an oncologist for baseline nerve excitability testing prior to commencement on chemotherapy. All study procedures should be approved by a hospital or institution internal review board. Screen patients to assure they have no history nor baseline neurophysiological evidence of peripheral neuropathy, that they have not received prior neurotoxic chemotherapy treatment, and have no contraindications.